So we're going to stay on the topic of mitigation, and our next speaker, Neil Wedlock, he's based at the Grasslands campus in Palmerston North. He's going to be talking about a vaccine that's been developed to reduce methane emissions from the ruminant animal.
Thank you. Good afternoon. Yes, I want to tell you a little bit about the work we're doing at AgResearch, developing a vaccine to reduce methane emissions in farmed ruminants, cattle, sheep and deer. This cartoon is a ruminant. It's going to be vaccinated. It's probably a sheep by the looks of it. And the vaccine works to stimulate the antibody responses in the components in the vaccine in the animal, crucially antibodies in the saliva of the ruminant. Now, ruminants produce copious quantities of saliva each and every day. A sheep can produce 16 litres of saliva. Cattle bees can be up to 200 litres of saliva. And most of that saliva is swallowed and ends up in the rumen. So once we vaccinate an animal against the methanogens that reside in the rumen and produce the methane, that is a sustainable effect. Every day the animal will be producing new quantities of saliva, new amounts of antibody that enter the rumen. Why do we need a vaccine? Why do we need a mitigation? Well, we've heard that agriculture contributes about 50% of the greenhouse gas emissions in New Zealand. And of that, the major component is methane, belched out by sheep, cattle and deer. In terms of a mitigation strategy, vaccines are safe, they can be made to be very effective. Crucially, they're acceptable to farmers and the general public. I'm sure many of us have had a recent flu vaccination, a very simple procedure, but the outcome can be very important. It's a low intervention system. Farmers are already familiar with using vaccines to control livestock diseases. So another vaccine would just fit in with their existing and very likely future farming systems. Now, you're probably wondering what that picture is. It's not a picture of a galaxy taken from the Hubble telescope. It's actually something much closer than that. It's actually, if you take a small amount of contents from a rumen, it could be a sheep, it could be a cattle, and you put that drop under the microscope. And remember, the rumen is a big fermentation vat. There's lots of microbes in there, lots of partially digested plant material. A lot of that material fluoresces, different colours under the microscope. And you can see those little blue dots. Those are the methanogens. They autofluoresce blue under the microscope. And if you take about a gram, about 5 mils of rumen material, there's about 5 billion of those methanogens. Each of them, or most of them, producing methane, and those are the organisms that we're targeting through a vaccine. There's a lot of different types of methanogens in the rumen, the large diversity, and only a few of them have been cultured. They're difficult to culture, they're anaerobes, any amount of oxygen will kill them, very fastidious in their culturing requirements, but a number have been cultured and sequenced. So the genome sequence is available, and that's been very important for providing information about how these organisms function, how they produce, what the pathways, the biological pathways, the biochemical pathways producing methane, and also has given us insights on how we can target these organisms through a vaccine. So we've looked at proteins that are expressed on the surface of the methanogens. These are proteins that have a normal cellular function, a critical cellular function. If we knock out the function of that protein by antibodies binding to the protein, then you'd expect the organism to be crippled, not to grow as well in the rumen, and certainly not produce the methane. We've vaccinated sheep and cattle with some of these proteins, and we've shown that they're immunogenic. The animals will produce antibodies against these methanogen proteins, produce antibodies in the saliva. And we've taken some of these antibodies and shown that they can inhibit the growth of methanogens in a test tube. So in a laboratory situation, we can grow these methanogens and we can add in some of these antibodies, and we can show that the antibodies will reduce the growth of the methanogens. And also there we can measure methane in the test tube as well. We can show that that's been reduced as well. So that's a first important step in terms of developing a vaccine. Now, these organisms, as I mentioned, are very diverse, but a number have been sequenced, as I said, and those are some of the names of these organisms, which I won't go into. But there's actually, although there's that diversity, there's actually two main groups of methanogens in the rumen, the blue and the red. And if you look at cattle and sheep and deer across New Zealand and look at the different diets they're on, that pie diagram varies slightly, but there's still those two major groups of methanogens. And if you look at ruminants across the world, cattle, deer, sheep, bison even, you find again some variations, but the same two groups are always present. And they're the majority of the methanogens. And those are the methanogens we're targeting with the vaccine. The main work we do is being funded by the PGGRC and Agricultural Greenhouse Gas Research Centre. But I will mention a project that is being funded by the Ministry of Primary Industries, and that's a project where we're looking at some of the more fundamental aspects of how antibodies bind to methanogen proteins. What happens to the antibodies when they go into the rumen? Do they get degraded? We believe they don't. So these are questions we have to answer to really develop an effective vaccine. And in that particular project, we have some partners, overseas partners in Argentina and also in Australia, and we have worked with University of Otago. So in summary, we can vaccinate animals like sheep and cattle to produce antibodies in the saliva against methanogens. We've identified a number of proteins that could be used to formulate a vaccine, and we're systematically testing these antigens to see how effective they are and find the best ones for making a vaccine. The vaccine is likely to be a mixture of different proteins. We want to be able to hit the methanogens hard, multiple targets. We're now at a stage where we're putting together some of these mixes into experimental vaccines, and we're vaccinating sheep, and after they've been vaccinated, we're putting the animals through the respiration chambers so we can measure the methane emissions. We can see whether the vaccine has reduced methane. We can also look in the rumen of the vaccinated animals. We can look and see whether the vaccine has changed the population of methanogens, has reduced the numbers of methanogens. Once we have the right mix, we have an experimental vaccine that consistently reduces methane in chambers. We will work with a commercial partner, an animal health company, to develop a vaccine that's suitable for New Zealand farmers. And because I said that there are two main groups of methanogens present in ruminants around the world, we believe that a vaccine that will work for the New Zealand situation will be universal and will also work worldwide. Thank you.